Hello and thanks for watching. This is Balancing Act Day 6. We're going to talk about the nitrogen cycle and some more experimental design. So just a quick reminder, you do have an assessment coming up in next week, so 4 or 5 days, and these are the class periods and the days. Another reminder that if we have a fire alarm during class at all, when that fire alarm goes off, please leave the room immediately. Remember to slide in your chair. We'll go silently and calmly down the hall. And whoever the last person is leaving, please turn off the light and close the door. And we close the door because it helps with some fire containment if there actually ever is a fire. All right, today, what am I learning? I'm going to practice what we learned about experimental design and experimental design diagrams. And I'm going to continue to look at the human impact on the nitrogen cycle. Why? This is because I want to understand how nitrogen moves through the ecosystems, both biotic and abiotic parts, and I want to be sure that I know how to set up an experiment. So right in the beginning of class, we asked people to take a look at the nitrogen travel log for human impact. That was page 7 of your packet, and compare it back to the natural impact that we could see on nitrogen. And so we did notice that with human impact, we could see that a lot of times the nitrogen was in fertilizer, it ended up in surface runoff, it ended up in soil. And of course, nitrogen still ends up in DNA and proteins in plants and animals. So I have a video for us to watch, but I'm actually going to maybe not do it during the video notes. This video is in your independent practice. So when you get to the independent practice, please go ahead and watch this video on acid rain. I will say to you that when humans burn fossil fuels or other living materials like trees, we call that biomass, those trees and things that might be burned. When humans burn those biomass items, there are some quantities of nitrogen that get into the atmosphere. They get into the atmosphere as nitrogen, right? But it's going to be a form called nitrogen oxides. That's not a form that we've talked about just yet. Acid rain is caused when these forms of nitrogen that are in the atmosphere mix with rainwater. Then they fall to the ground, and they're going to cause some nitrogen pollution in waterways, and they're going to cause acidic water in our waterways. Okay, we want to understand that most companies have created ways to minimize how much nitrogen enters the atmosphere, especially at least in the US and Canada and Europe. So make sure when you get to your independent practice that you do watch this video. All right, more reminders about things we're learning about. A science class is creating aluminum foiled boats in various shapes. We even have the word various, which has part of variable in it, right? Pennies will be added to each boat to determine how many pennies can be added to the boat before it sinks. So this is the experiment. We have different people making these different types of boats. What is the independent variable then? What's the one thing the science class will change? It's the shape of the boat. And then they want to know what effect did the shape of the boat have on how much it can carry. So they're going to measure their dependent variable. They're going to measure how many coins they could put in there, how many pennies. Okay, in another example, Professor Sprout wants to see if adding different amounts of fertilizer or nutrients will help her mandrake plants grow larger. So what is the independent variable? What's the one thing Professor Sprout changed? Let's look back up here. Professor Sprout wants to see if adding different amounts of fertilizer. So she's thinking about changing the amount of fertilizer, right? She wants to know, will it help her mandrake plants grow larger? So I'm going to have to measure the size of the mandrake plant. That's my dependent variable. Measure plant height, right? All right, so another reminder, and it's a good idea to have your packet out. You're going to want to be filling in information from your independent work in your packet, as well as doing slides. Right on the front page of our Balancing Act packet, it does say that in this investigation, we will conduct an experiment to determine the effect of nitrogen on aquatic plant growth and water quality. in aquatic ecosystems. We will be doing that starting next week. We will actually do some more ecosystems, and we'll be doing water quality testing. Okay, what are these? These are vitamins. Why do people take them? People take them so that they can have a better balance of vitamins and minerals and hopefully be healthier, right? Is more better? You might remember from being little, if you had vitamins like this, they taste yummy. And so you might have asked your mom, can I have another one? And hopefully your mom said no, because too many can actually be harmful. So there's a specific amount that is just right for your body, and more is not always better. So what are these? Pour and feed, all-purpose, fertilizer, and plant food. They're all types of plant foods or fertilizers. Okay. Why are they used? What is their purpose? There's a reason I have these right after Flintstone's vitamins. These are basically vitamins and nutrients for plants, just like Flintstone's vitamins would be vitamins and nutrients and minerals for humans. Okay. So these are things that are going to help the plants grow. Again, our job is that we're going to conduct an experiment to determine the effect of nitrogen on aquatic plant growth and on water quality. What we want to realize is that nitrogen is one of the main components of our fertilizer. So we're going to use fertilizer to find out what's the effect of nitrogen. So everybody got a recording sheet. The recording sheet and the different around the room scenarios. There are 7 of them. It's posted in Schoology, so if you're not in the building, you can look on Schoology and see the different scenarios. I gave everyone in class 12 minutes. I'm not going to go 12 minutes on this video. But here was number 1. Think all the way back to the first lab. And please do look at the link that is in Schoology, or make sure you've done the scenarios in the classroom, because they have more information for you. This is my answer section, so after you do those on your own, this is my quick set of answers. Okay, so in our ecosystems, we had put in Chlamydomonas and Daphnia, and we put in Spirogyra, and we put in Alodia, duckweed, guppies, fiddler crabs, pond snails, and black worms. Those were the items that we put into our ecosystems. So hopefully you remembered some of those, and a couple people remember all of them. Then the second scenario or station asked you to think back to the aquatic ecosystems again. What were the 5 water quality measurements that we did? And so you were writing this down on your recording sheet. What were the 5 things we did? We tested nitrites, we tested pH, we tested dissolved oxygen, and temperature and clarity, water clarity. Then Station 3, our next job is to design an experiment to see how nitrogen, which you get from fertilizer, impacts plant growth and water quality. Okay, if I want to do an experiment, what materials might I need? So remember, you want to make sure you try to answer this yourself first, then check your work. We're going to need fertilizer. We're going to need a plant, right? We are going to use Alodia in the classroom. We're going to want some Chlamydomonas or algae, because that's going to be part of the experiment in our design. We're going to use a jar to contain these things. We'll need some temperature probes and pH probes and dissolved oxygen probes. And we're going to use our nitrite testing system again. All right. So again, in the next experiment, we're going to check the effects of nitrogen. Remember, that's nitrogen in fertilizer on plant growth in an aquatic ecosystem. So which organism from our aquatic ecosystems in the past would be best? We've decided Alodia is going to be the best for us to use in this experiment. We're designing the experiment to test the effect of nitrogen, which is a nutrient, on plant growth and water quality. So which water quality measurement do we think would be really low if we didn't have plants? Notice these bubbles in the image here. Those bubbles are actually oxygen bubbles, because oxygen is being released from those plants. So if we don't have a lot of plants, we're going to have a really low dissolved oxygen. Okay, because plants produce oxygen when they do photosynthesis. So we're thinking back about our photosynthesis. On your recording sheet, I asked you to write down the equation for photosynthesis and try to remember amounts. So you could write it with the words or with the chemical equation. I'll show you both. Our word equation is we have carbon dioxide plus water in the presence of light in the chlorophyll produces glucose and oxygen. So we want to know how much of each one, right? And I always remember 6, 6, 6, and 1. One of the big giant honker glucose molecule. Because remember with our poppet beads, that was a big giant set of things stuck together compared to everything else. So that just 1. Everybody else gets 6. 6 carbon dioxides plus 6 waters in the presence of energy and in chlorophyll produces 1 single glucose and 6 oxygens. All right, and again, what is the main ingredient in plant fertilizer? Well, it's nitrogen. And so if we want to think about 2 things we already know about nitrogen, we know that it is in plant food, so it's something that we want to have for our plants. We also know that if you think back to Watershed Walk, we know that's one of our biggest pollutants that gets into surface runoff and ends up back in the Chesapeake Bay, right? So too much of this fabulous thing is actually not a good thing. All right. The main ingredient, again, in fertilizer is nitrogen. And so the right amount of nitrogen should make plants healthier. But too much actually might be a bad thing. Nitrogen is a nutrient that we need when we have plant growth. So a lot of times you'll get a container like this that has liquid inside. The liquid is made up of lots of stuff, but you can see that it specifically has nitrogen listed as its first ingredient. Okay. All right. So after you complete your recording sheet, turn that in. If you were not in the building, make sure you turn that in next time you come to the classroom. Make sure your first and last names are on there. All right. So our experiment, we are looking at an experiment to determine the effect of nitrogen on aquatic plant growth. Again, I'm talking about that. So there's a packet for everybody in the classroom. I also posted that packet in Schoology, and it is called Balancing Act Fertilizer Experiment or something. Hopefully it'll say fertilizer packet. I don't remember the full words. So as we looked at that packet, we started filling out what we could. So we're going to come up with our experimental design steps. The first thing we need to do is have a question. What's our question? Our question is, how do you think nitrogen, which is a nutrient, in fertilizer will affect or impact plant growth? Again, we're going to be using Miracle-Gro, I believe. Then if we want to design an experiment, of course, we want to keep it fair and valid. We want to be sure we're measuring what we say we're measuring. Okay, and we need to design the experiment, so we're going to need to know what materials we have available to us. The materials you can use are fertilizer, Alodia, Chlamydomonas, which is an algae. We're going to have 3 mason jars per station because we're going to have 3 levels of the independent variable. These are all of our probes, the temperature and dissolved oxygen and pH, and then we'll have nitrite tests. So write down all of those things, please, in your materials section. Based upon what we know, I'd like for you to complete the experimental design diagram with a partner if you're in the classroom. If you're at home, maybe you can Google chat or text with a friend, or even see a friend or talk to them in person. And we're going to try to fill out as much of this page as we can. Go ahead and do that in pencil, and then you can check your work because I'll go over what I think goes in each section. Okay. The independent variable is the amount of fertilizer. We are measuring the nitrogen, and by that I mean we are going to we are changing the amount of nitrogen because we are changing the amount of fertilizer that's going in. The levels that we're going to use are 0 milliliters in one jar, 6 milliliters in another jar, and 18 milliliters in the third jar. We have 8 stations for 8 student groups, just like we did in our original ecosystems. So we're going to have 8 trials for each level, okay? 8 jars that have 0 mils, 8 jars that have 6 mils, and 8 jars that have 18 mils. Remember, with milliliter, you do write it lowercase m, capital L. Liter is always capital L. All right, so for a control group, we always want to think, what are we going to look back and compare back to? We're going to compare back to putting no fertilizer in. That's going to be our control group right there. And the dependent variable is the thing we're going to measure. This one, actually, we have a lot going on. We're going to measure water quality, but also plant growth. So you can write on your page plant growth and water quality. If you want to specify water quality, we're going to check water color or clarity. We're going to check temperature, dissolved oxygen, nitrites, and pH. The water color or clarity is going to actually give us an indication of how much algae growth is happening. All right, and then constants, we have the same size jar for each. We have the same amount of water for each, the same type of plant, Alodia, and the same amount of plants, okay? We're going to have the same basic starting size of plants so that I don't have one jar jammed all full of Alodia and another with just a sprig. We're going to try to be pretty similar. We're going to keep everything at the same temperature and have the same amount of sunlight. We will use the same type of fertilizer, and we will control the amount of algae we are adding. That's going to be a constant. If we have to write a title, we might say the effect of the amount of fertilizer, or nitrogen, on plant growth and water quality. If the title you wrote is slightly different, as long as it says the same basic thing, that's great. All right, and then my hypothesis could be if more fertilizer is added, so if more nitrogen is added, then the plants will, I'm going to say grow less if it's me, and the water quality will, I have to decide, is it going to increase or decrease? Some students said increase, some students said decrease, so you will choose and write that down on your paper. All right, then we need to create a data table or a spreadsheet. In your packet, I've actually done that for you. So this is the type of data we're going to be collecting starting next week, and this data table already exists in your packet. Okay, and then we'll collect the data and we will analyze it. So I gave everybody a moment to look at some dad jokes. I'm not going to do those in my video. That was a little brain break. And we want to make sure we're understanding that humans impact the nitrogen cycle. So in Schoology, please find the link that asks you to go through a Google Slides assignment. Just before I had people get into that link, I asked everybody to just take a look. On this map, any one of these red markers indicates something called a dead zone. And so I asked people to make some observations about where these red markers are, and think about what questions you have. In class, most people realize that these are right along oceans or major water. People notice that on the east coast of the U.S. and up over here, perhaps there's more people living in those areas. So you are thinking about that because we will revisit this next class right before our assessment. All right. You're going to get into Schoology, into the assignment that says human impact on nitrogen cycle. And we did watch this video if you were here in class. Otherwise, you're going to watch it when you're doing your slides. So go to Schoology and find the independent work. Other things you need to finish today, you're going to do the independent work. You're going to make sure you're finishing up your packet because we're nearly done with your packet. We have an exit ticket for today. And then I have posted a study guide on Schoology that has a lot of interactive links. So I didn't actually give that out on paper. Make sure you get in and try some of those study guide items so that you are really set for your upcoming test. Okay, after you do your human impact, you can come back and look at what I think the answers are for some of your human impact. Do that first, so you're going to have to pause this video and come back. Okay, so hopefully you're all done with your independent work. And this was one of your first questions. What living thing is in the water that makes it turn green like this? It is algae that's making it turn green like that. Then, what happens when algae gets a lot of nutrients? Well, it grows and grows and grows and grows and grows. It grows, it gets really happy and grows. When we have too many nutrients in the water, so you see how this was a part of a river, I believe, not very fast moving. We get a really thick covering of that algae. We call these algae blooms or algal blooms. It's a really big, thick covering, fast growing algae. Look at that. Ooh, delicious. Okay, so why are algae blooms a problem? They're a problem because they block sunlight that the stuff underneath the algae really needs in order to grow. Okay, we need our stuff underneath the surface to grow and photosynthesize in the sun because that puts oxygen into the water, yeah? Yeah. Okay, what underwater needs that sunlight? It's called submerged aquatic vegetation, SAV. That just means underwater plants. Okay, when those plants can't get their sunlight, they're going to die, right? Because they need to do photosynthesis because that's actually going to allow them to produce glucose or sugar that they need for their processes. So if they can't do it, they're going to die. All right, when the submerged aquatic vegetation dies, now they're not doing photosynthesis, so we are reducing the amount of oxygen, the dissolved oxygen in the water. Those dissolved oxygen levels drop. That means that animals lose their habitats and food, so they're going to move away or die. Poor fish. And what happens to those dead organisms? Well, typically they are decomposed, right? Decomposers would break them down, but decomposers need oxygen or else they can't do their processes. So the more dissolved oxygen you lose, we actually have less of an ability for those decomposers to break them down. So when there's no more dissolved oxygen in a section of the water, it is going to be called a dead zone. You can pause the video here and sort of read some of these little captions. The process in which nutrients cause those algae blooms, which are the things that cause the dead zones, this process is called eutrophication. Eutrophication. All right, and so let's think back. How do humans impact the nitrogen cycle? Well, we apply excess fertilizer, which runs off in the surface water. We are combusting a lot of fossil fuels, and that actually can impact the atmosphere, and we end up with some acid rain, right? And also because we have more animals that we are creating and growing and keeping alive on farms and things, we end up with even more animal waste that ends up in the surface water. The result is that we have too many nutrients in the waterways sometimes and too many nitrogen oxides in the atmosphere, which causes acid rain. All right, so thanks so much for watching. Please go through some of those, possibly all of those links that are in your study guide. Try to make sure that you've got your packet all filled out and do the exit ticket for day 6. And before we actually take our assessment on day 7, we will have some review. Thank you so much for watching.